again, Avian Adenovirus. In this lecture, we will speak about the avian adenovirus infections, common in poultry, and some can be defined in terms of clinical and pathological characteristics. The group of diseases affect poultry. Adenovirus play primary or secondary role in a variety of syndromes, including inclusion body hepatitis and hepatitis hydropericardium syndrome in chicken, hemorrhagic arthritis of the turkey, egg production decline in laying chickens, egg drop syndrome 1976, bronchitis in quail, and other respiratory arthritis, encephalitis, and enteric syndrome including gizzard erosion, pancreatitis, and tuberculitis. History: the first recognized adenovirus infection of birds with quail bronchitis described in 1951. The etiology of adenovirus are DNA virus that replicate and produce inclusion bodies in the nuclei of the infected cell. Inclusion body inside the nuclei. Adenovirus classification is not intuitive. The family Adenoviridae is divided into 5 genera, and adenovirus infection in birds are in 3 genera: group 1, group 2, and group 3. And we found adenovirus affect ducks and pigeons and turkey. The epidemiology: adenovirus can be transmitted both by vertical and horizontal. Clinical signs: disease with well-established adenovirus etiology, namely inclusion hepatitis and hemorrhagic arthritis and egg drop syndrome 1976 and quail bronchitis. Diagnosis: routine diagnosis of infection is typically carried out by the combination of virus isolation, post-mortem examination, histopathology, sometimes electron microscope, or polymerase chain reaction. Inclusion body hepatitis. Inclusion body hepatitis is a disease of young chickens characterized by sudden onset, increased mortality, and hepatitis accompanied with intranuclear inclusion bodies. In 1963, hepatitis with inclusion bodies was described in chicken, but the causative agent was not identified. In 1970, a similar disease occurred in many flocks in Canada and the United States. Adenovirus was isolated from an Indiana outbreak, and eventually from flocks in many other locations. Inclusion body hepatitis can be transmitted both vertically and horizontally. In exposed birds, the virus enters via the alimentary tract, and in some cases by the conjunctiva nasal passage. Primary replication occurs in the nasopharynx. Egg-transmitted adenovirus may remain inactive in infected chicken or poulets until maternal antibodies wanes. Outbreak in young poults 1 to 2 weeks are typically associated with vertical transmission, while outbreak in older birds are most often due to horizontal transmission. When broiler from multiple sources are mixed in, it's sometimes impossible to determine the source of infection. Clinical signs: a sudden marked increase in mortality is often the first indication of the disease. Mortality increase for 3 to 5 days, level off for 3 to 5 days, and then decrease to normal level over another 3 to 5 days. Total mortality may be as high as 30%, but is typically considered lower. There are few specific signs. There may be pallor of the comb, wattle, and facial skin. Infected birds are depressed and listless. The lesion, petechial hemorrhage, and ecchymotic hemorrhage may be present in the skeletal muscles. The skin of the inclusion body hepatitis. Chicken are markedly swollen and pale and mottled. At microscopic presence at microscopy, presence of the multifocal to local extensive degeneration necrosis of the hepatocytes, pancreas of necrotic spots, and congestion are visible on the surface of the organ. Microscopically characteristic large basophilic intranuclear inclusion polyadenovirus inclusions in the hepatocyte are observed. Erosion polyhepatitis in chicken, the liver is swollen, enlarged, then with mottling with petechial hemorrhage under the capsule in the parenchyma. Diagnosis in young growing flocks, sudden increase in mortality is suggested to erosion polyhepatitis. Histopathology, investigation of clinical microscopic lesion in the liver, including the characteristic intranuclear inclusion bodies. Virus isolation of the virus, PCR detection, serology, seroconversion to inclusion polyhepatitis associated serotypes, genotyping, analysis of the nucleotide sequence encoding adenovirus, control license vaccine against the disease. Kill pathogenous vaccines has been used. Treatment, no treatment. Only supportive treatment can do.